Good evening, everyone. I'm Andy McMahon. I'm the director of the Eli and Edith Broad Center for Regenerative Medicine and Stem Cell Research, which is this building here. I want to give you three pieces to put this meeting today in context. In 2004, when I was still in Massachusetts, Californians made the very sensible decision to set aside $3 billion of taxpayers' money to set up the California Institute of Regenerative Medicine, broadly to bring stem cell-based therapies to the clinic. The second piece of context has to do with the actual subject that we're dealing with today, which is the problem, the most common form of blindness in the aging population, which is age-related macular degeneration, and our mission to try and give the public a sense of what it takes to take research all the way to pioneering therapeutic clinical trials. The third piece of context is today, the International Society for Stem Cell Research, which is the largest world organization of stem cell scientists, is in Los Angeles. There's 4,000 to 5,000 scientists at the convention center, and we wanted to celebrate the fact that the world is looking at Los Angeles today for this public outreach forum. This is designed to be a forum where it's an interchange between the scientists and clinicians and the public. And the way we're going to do this is we're going to take you all the way through from discovery research to the clinic and then have a question and answers forum. I just want to point out a few people who are very important to tonight's proceedings. And I'll ask them to stand and wave something so we know who they are, so that you know to single them out. So we have John Thomas here. John Thomas is the chairman of CIRM, the California Institute of Regenerative Medicine. That's the organization that you wisely put your money into. We have Melissa King. Melissa King is spearheading America for Cures, which is going to bring and is bringing the case for what this has actually done to put California at the front line of stem cell research worldwide. And then our Masters of Ceremony is Kevin McCormack, who is the face, the public face of CIRM. And he's much more eloquent and much more convivial than me, so I'm going to hand over to Kevin right now. Thank you very much for coming here.
Thank you, Andy. Thinking of me as the public face of anything is a rather scary thought. Andy has a number of remarkable attributes, one of which is that he has something in common with Sir Isaac Newton, the fellow who just dropped an apple and discovered gravity, and Charles Darwin, the fellow who basically realized we're all just basically apes. And that he is a member of the Royal Society of London, which is a very distinguished group of people, scientists, some of the best scientists that have ever been around. So we're in a distinguished company here. And the motto for the Royal Society is nullius in verba, which loosely translated says, take nobody's word for it, which I think is really good advice to any scientist. And I'm sure most of the scientists here started out with someone saying, you can't do that. And they didn't take their word for it, and they did it. And a lot of what we've been funding at CIRM over the last 15 years is scientists like this, scientists who didn't take anyone's word that you couldn't do that kind of work, that wouldn't work, that it was impossible. And in the last 15 years, we have now funded 55 clinical trials, and everything from HIV to heart disease and stroke, spinal cord injury, and a number of in vision loss. And that's what we're going to hear about tonight. And the story you hear is remarkable in many ways, in that it's painstakingly it's painstaking science to kind of build this up one step at a time, and then to try and get the funding for it, and to try and do so many things. And the small part that we were able to play at CIRM is that we were able to provide that funding thanks to you, the voters of California, and to you who are going to be the next voters of California. So we're thanking you in advance. It created a possibility of funding groundbreaking research that otherwise might not have been possible. That at a time when the federal government was cutting back funding and putting restrictions on funding for stem cell research, the people of California said, this is crazy, this is too important. And so we were able to, they created us, and we were able to kind of help fund the research that you're going to be hearing an awful lot about tonight. So we're going to start with Dr. Mark Humayun. Mark invented a retinal prosthesis to help people with vision loss, but then he decided to go one better and create something that would help people who are suffering from macular degeneration, the leading cause of vision loss in this country. So here's Mark.
Thank you, Kevin, for that introduction, and welcome to all of you. We're having just great weather here, and we'll try to, hopefully you can see the slides and some of the graphics we're going to present. We've kept the graphics to a minimum, realizing we're outside, so it's going to be mostly talking through the science. But again, if anybody's interested to want to learn more, we're happy to provide more details. So as Kevin just mentioned, my name is Mark Humayun, and I'm an ophthalmologist and a biomedical engineer. And I would like to thank Andy McMahon for inviting us to showcase what we're doing. So my grandmother went blind from diabetic retinopathy, and is what got me interested in ophthalmology. And from there on, I've been working very hard to try to restore sight to those who are blind. And this is a situation we're in with macular degeneration. Unfortunately, many of the patients end up with blindness. Now, there are different types of blindness, and in this condition, what happens is you end up losing the very central vision. So you can't read, you can't drive, you can't recognize faces, you can't watch television. And it occurs starting could be as early as in the late 50s, but really in the 60s and 70s. So we all work very hard. We look forward to those golden years, and unfortunately, everything is working on you, your heart, but you're robbed of your vision, and it takes away your independence. So a devastating disease. There are certain treatments we can do for the wet type. I'll explain the different types, but for the dry type, which we're attacking with this technology, there is currently no treatment. So you see this graphic up here that we're showing. And for those of you who are not familiar with the eyeball, this is looking inside the eye. We can look inside, and you can see there's this, hopefully you can visualize it. There's this, looks like a bottle-shaped device or paddle-shaped device. That's the implant with the stem cells, and it's placed in the very center underneath your retina to help restore and regain the vision in the very center. So I'm up here to introduce our team. Many of them are here, and I'm very grateful for their hard work. We're going to talk about a little bit about the technology, and now next. Clearly, this would not have been possible if it wasn't for the generous funding of CIRM, and on those of you who voted for Proposition 71 to allow that money to be put towards CIRM. And these projects take a lot of money. Just, if you're starting with an idea and to get it into patient, it can take millions of dollars, tens of millions of dollars. So it isn't something that is a few $100,000 or even $1 million. It takes a lot of dedication, a lot of time, and a lot of belief in these technologies. So as I mentioned here, the technology, this will a little bit more about the disease. As I mentioned in macular degeneration, again, you can see on these slides, if you look in normal vision, you can see the two children with the balls. but in macular degeneration, wherever you look, in the very center, you cannot see. So for example, in this case, if you're trying to see the face of those two kids, you cannot see that. There's a blind spot. So there are two types of macular degeneration that occur. One is the bleeding type. We call it the wet macular degeneration. It's about 10 or 20% of patients with macular degeneration. And for these, we do injections. And if they get really very severe, there's an effort in England that is helping develop a stem cell approach for these as well. Our technology could also potentially work for this condition, but we've really focused initially on the dry macular degeneration, which is about 80 to 90% of macular degeneration. And in this case, it's an aging condition and you lose a certain cell type in the eye. But not only do you lose the certain cell type in the eye, but you also lose the membrane that these cells sit on. So in layman's term, you've not only don't have seeds to plant, but also the soil where you need to put the seeds is also not good. So you need to create a membrane to put these cells on and then get it underneath the retina. So that was what we were faced with. And here is the slide. So after 5 years, a long period, we were able to accomplish this and put the cells such that they can be on a membrane that is biocompatible and it breathes and lives like the normal membrane that is in that area in your eye naturally. And these cells derived from embryonic stem cells, they're RBE cells, line up perfectly, one cell at a time, and they're oriented, meaning the top side being top and the bottom side being bottom. It's really important. If you don't get these cells to line up in a single layer, if you don't get them to be pointed the right way, well, then they don't function very well. So this membrane allowed us to carry these cells. We differentiated the cells. So now you have the combination, both, so to speak, the soil and the seed. And so we were able to implant this, and the end result is, as you saw at the very beginning, that picture that you saw in the retina. So this is truly an interdisciplinary convergence, and that went by way too fast, so I'll back it up. So to make this sort of thing happen, it's just not one discipline, and it's often we deal with this situation. It's very difficult. If you know biology, you feel very comfortable in biology. You do not want to be talking to a material scientist. In fact, if you try, it almost gives you a headache because you're speaking two different languages. So, but in order to make these large projects work, you have to work in interdisciplinary teams and develop this convergence. So this is, for example, neuroscience, pharmacology, neuroplasticity, to just name physiology. Not psychophysics is a way in the clinic where we test how much vision the patient does gain. So in any event, it's the convergence of all of these that come together that allow us to work together. So the multidisciplinary team that is going to be coming up and speaking with you, to you and with you about this implant stems from a number of different universities. So we have Dennis Clegg from University of California, Santa Barbara, who will be speaking shortly. Then we also have David Hinton from the University of Southern California, as well as Amir Kashani from University of Southern California. We also have Joe Gould from the City of Hope. A very important aspect of developing these technologies is you need someplace to manufacture it. You can't do it in your lab and put it into a patient. It needs to be regulated. And so there's these good manufacturing practices that are very important. And City of Hope played that role for us. And then, of course, Jane Lebkowski from Regenerative Patch Technologies, which is the company that's commercializing this technology, will speak to you about how do you navigate the regulatory FDA and how do you get it in trial and how do you move it forward. So hopefully in this brief introduction, I've given you a little bit about the disease, what we're trying to address, B, about the technology that is just embryonic stem cells on a substrate. We need both. And C, that really is an interdisciplinary convergence and it takes a number of different areas. And I do want to also point out Caltech, who was instrumental in helping us develop the membrane, because the membrane is made much like a computer chip is manufactured. It uses photolithography, this process, so it isn't a straightforward process, but it's a process that we know that works very well for computer chip technology. So with that, again, I'd like to acknowledge CIRM for its funding and Andy McMahon for inviting me, but we'll move to the next presenter. Dennis, if you could come up. We'll take questions at the end after all the speakers have presented. So, Dennis Clegg.
All right. Thank you, Mark, and thank you all for coming. It's a beautiful evening here, and thank you for coming to learn about what we're doing and really learn about what maybe the future of medicine will be. Certainly a big part of it, as we try to treat all these diseases that currently cannot be treated. I'm Dennis Clegg. I'm from UC Santa Barbara, just up the coast, where we don't have a medical school, but we do a lot of biological research and biomedical research. And we started this project and wanted to find the world's dream team, the best people that could bring this to patients. And so Mark talked about this team that we put together, and with funding from CIRM, moved this from bench to bedside, as they say. Our part at Santa Barbara was to figure out how to make the cell type that we need, the RPE, retinal pigmented epithelium. If you look into someone's eye and you see the pupil, it's black. That's not because it's dark inside the eye, it's because there's this monolayer, single layer of pigmented cells in the back of your retina that are crucial for support of your retina. And macular degeneration is caused by the death of those RPE cells. So can we make cells and replace the ones that are dying in patients? And we started with these cells, if you can see the next figure here, human embryonic stem cells. There are a lot of different kinds of stem cells, but human embryonic stem cells are the most powerful. And we turn to the most powerful cell type because this is such a serious disease that currently doesn't have any treatments, no treatments available. These cells come from leftovers from in vitro fertilization, where instead of throwing them away, couples that have made this choice to do in vitro donate these to science. And this cell line that we use was generated in Wisconsin in 1998. And it's so powerful, it can make every cell in your body. There are 220 some different cells. You've got bone cells, brain cells, and eye cells. We wanted to specifically make one, the RPE. We didn't want to make bone. You don't want to put a bone cell in your eye. That would be bad. You don't want to put a heart cell in your eye. You want to make specifically the cell you need. And so my lab figured out, if you go to the next couple slides, please, the next couple figures, we wanted to convince them to become RPE. And we worked for many months and years to add the right growth media to make them into RPE. And then, as Mark said, Caltech worked to provide a scaffold to grow the cells on. Imagine, how many wear contact lenses out there? Anybody? Okay, I do. Imagine a little tiny piece of contact lens-like plastic material, and you grow cells on top of that, but instead of putting it on the surface of your eye, the surgeons, the talented professionals here and eventually other places, actually transplant that into the back of the eye to replace cells that are missing. And Mark showed pictures, if you go to the next couple figures, of our implant. And I'm showing her a picture of the patch, we call it, under the microscope, where if you zoom in at the lower figure, you can see high magnification views of these individual cells as a monolayer that are on the patch. We developed a surgical technique that you'll hear about to implant this in the eye. And we're in the middle of a phase 1 clinical trial, which you'll hear more about in a minute, where we've actually implanted this into patients to treat them in an experimental clinical phase 1 trial here at USC. And we're hoping to get this out to the millions of patients in the world that suffer from this disease and treat their unmet medical need. So that's our part of the project, and I'll turn it over to the next speaker, Dr. David Hinton, who's local from here at USC. Thank you very much.
Hello, everyone. It's really nice to be here. I'm David Hinton, professor of pathology and ophthalmology at Keck School of Medicine. In fact, my whole career has been at the Keck School. I trained as a neuropathologist, and I've been studying the pathogenesis of retinal disease for over 25 years. And also, I've worked on this project for just over 15 years. So first of all, I had a foundation grant, which allowed to get some preliminary data. And then I got a CIRM seed grant, which allowed us to get even more preliminary data. And then we got the CIRM disease team grants, which have allowed us to progress so far. And that's where we've been able to show the tremendous interdisciplinary research involving all of these groups. So once the stem cell-derived RPE implant was developed, as you saw, there were many preclinical studies that had to be performed. And many of these are done in rats to demonstrate safety first and then efficacy or function. And this culminates in extensive supporting documents that then are used to enable Food and Drug Administration clearance to go ahead with clinical trials in humans. So we did a lot of safety studies. Many of them were done in culture dishes, so we call that in vitro studies. But I'm going to show you a little bit about the studies that were done in rats. So the safety studies can include determining whether the implantation procedure is safe, whether the implant elicits an inflammatory response, whether the cells remain at the site of implantation, and whether the cells remain as differentiated RPE cells and don't proliferate or form a tumor. That was at one point a major concern in patient trials, but using these differentiated RPE cells, no one has ever found any indication of tumor formation, so they're very safe. So all of these studies showed that the implant is safe. Efficacy studies show that the implant is able to modify the course of disease in an animal model of retinal degeneration. And most of our experiments were performed in the RCS rat, which is a rat that has a genetic defect in the RPE cells that results in a retinal degeneration. So it was a very good model to use because the primary defect in AMD is in the RPE cells. And that's so very similar in the RCS rat. So I'm going to show you an example of one of these efficacy studies. So who can tell me which side is better to show the slide on? This one? Okay. I'm a pathologist, so I can't help but show you some histology sections because that's what I'm expert in. So this is the... No, I think it's the speaker. Okay, is that better? Yeah. Okay, so this is the normal rat retina. So in the eye, the light comes this way and then focuses on these light-sensitive photoreceptors, and that's where the signal for vision occurs. Then these cells then interact with inner retinal cells, and then they interact with these cells towards the surface of the retina, and their processes form the optic nerve that goes to the brain, where the images can be processed. So this is the area where AMD occurs, in this outer part of the retina. So I want to compare that with an RCS rat, and if you look here, you can see the retina is much thinner, and these inner retinal cells look pretty much the same as in the normal rat. But the outer retinal cells, these photoreceptors, are almost entirely gone. You really don't even see any cells here. So the RCS rat gets a very severe retinal degeneration of the photoreceptor cells because the RP cells are not functioning. So then we would put in our implant, and it turned out to be so interesting. So here is that paralleline membrane. These are the human stem cell derived RPE cells, which were implanted under the rat retina. And now this is rat retina, and you can see all the photoreceptors. This is at the same age as the model here. So all of these photoreceptors were preserved. And so these human RPE cells were functioning to help the survival and rescue of the photoreceptor cells. And these animals also were able to show had better vision. So it was with experiments like this and many more that we were able to provide the data to get the FDA clearance and then move on to clinical trials. Okay. So our next speaker is Joe Gold from City of Hope.
All right, well, thank you. It's a pleasure to be here tonight. My job title is Director of Manufacturing at City of Hope, which sounds like a strange job title for someone who works at a cancer center. But that's because I'm the director of the Center for Biomedicine and Genetics there, which is a 20,000 square foot manufacturing facility where we make things that range from the specialized viruses to reprogram a patient's T cells to fight their cancer to novel cell therapies for diseases like heart failure or ALS, or in this case, macular degeneration. Now, when you look at the kind of work that we do, you'll find that it differs from the sort of work that goes on in a typical research lab because of three letters, GMP, which, as we heard, stands for Good Manufacturing Practices. And GMP really is an all-encompassing system to make sure we do things right. It's very demanding. FDA requires it, and most places don't even attempt it because it's so hard. But City of Hope has 3 GMP facilities. Now, what does it really mean to apply GMP to cell therapy manufacturing? Well, what it really means is that we start by being very careful about the raw materials we choose. Are they suitable to put into human beings? They're not the same thing as research materials you might use in a typical lab. And once we've found those materials, we have to literally investigate the suppliers to make sure they're making them properly. When we get those materials into our facility, we control their use very carefully so that we know exactly when they were used and where they were used so we can trace everything. And then we perform some pretty complicated procedures to make the actual product that each step has to be verified and recorded. And then finally, we test the final product very, very extensively before it goes to patients. So if you look at the kind of pictures I have up here, and hopefully they're there, I can't see them, but one of the things you'll see that's different, if you look in our labs, you see how elaborately gowned our staff are. Well, that's partly, of course, to protect them from the product they're working on, but really it's mostly to protect the product from them because we have to keep out every type of contamination, every type of virus or bacterium or dust or microscopic particle. And you'll also notice you will never see just one person working in a GMP suite. And that's because GMP requires that we always have one highly skilled person performing these complicated operations, but one person literally standing behind them with a checklist, making sure every step has been done properly and that every calculation was done appropriately. And it would not surprise you to learn that we control access, who gets into our facility, but once people are actually in our facility, the people who are actually doing the manufacturing, they have to follow very, very prescribed routes of how they go in and out of the clean rooms, because the goal is to make sure there's minimal chance of contamination of 2 different products, or even to have 2 different production teams mixing together to avoid any types of contamination there. So any GMP facility that's successful really is successful because of a sort of dynamic tension, if you will, between two independent groups, which is the manufacturing group, which I head up, and the quality group. And they're it's a checks and balances system, and they have to be completely independent to make sure that we're all doing this honestly in this case. So how did this all really apply to the therapy we're making here? Well, what this really meant was that first, the scientists who you saw who developed this process transferred it to us, to my teams, and then my teams spent time to make a specialized master cell bank of the starting embryonic stem cells. So this bank was made, it's hundreds of vials of frozen cells, and then it was tested very, very extensively, dozens of tests to make sure that it was sterile and clean and fit for purpose. And then the next step was to thaw some cells from that bank and work with them in culture for about 7 months to grow them under the right culture conditions that they became partway differentiated to the RPE cells we wanted, at which point those cells were harvested and frozen into an intermediate cell bank, which underwent the same extensive testing as the first bank. And then, when it was time to actually make the product for the clinical trial, we would thaw cells from that intermediate cell bank, put it on this very fragile synthetic membrane, and then carry out tissue culture for another 50 days, which involved a lot of complex fine manipulations and a lot of observation of each implant under the microscope to make sure none of them had any defects. And the end result, after a whole lot of testing to make sure everything was sterile and everything was expressing the right genes to make sure they're RPE cells and to make sure we couldn't detect any signs of any remaining undifferentiated stem cells that might cause a tumor, that's when we could send them to Dr. Kashani, who'll tell you about how he actually had to put them in the eye. So I think you can see from this that the manufacturing of the cells may not be the most glamorous part of this whole process. In fact, it can be very slow and painstaking and tedious. But the reason you do it right is to make sure you come up with the kind of therapy we're talking about here tonight, which is as safe and efficacious as we know how to make it. So from that, I'll turn it over to Amir.
Good evening, everybody. Thank you all for coming out. My name is Amir Kashani. I'm an assistant professor of clinical ophthalmology. What that means is that I see patients in the clinic who have problems with their eyes and specifically with their retinas. So problems that some of you are probably familiar with or family members, such as diabetic retinopathy, such as macular degeneration, such as retinal detachments. And so I came into this picture after all these talented people had performed more than a decade of work to find out how to make the perylene membrane as thin as it needed to be so that the cells could grow on it, to find out how to make the stem cells into the RPE cells so that we can have cells to replace the ones that died, to find out how those RPE cells would function and survive and behave when they were in the place. So I hope all of you can appreciate and all of us can appreciate the tremendous amount of work and talent and science, basic science, that has been put into making the little patch that Mark showed you earlier and that's on that screen. It's not just something that we made even in 10 years. It's a result of a lot of laborious basic science. And my hat is off to CIRM for funding the basic science and the translational science that puts things into clinical trials. So all of this basic science that you've heard about, translating that into the clinic is a science in and of itself. And that's the science of clinical trials. And that's what we were charged to do. So we had to develop a surgical method to, as safely as possible, insert this implant into patients' eyes who are suffering from severe vision loss and age-related macular degeneration. And we had to select the people who would be most appropriate for that clinical trial. So 50% of the battle is really the science and the methodology. But the other 50% are the people, the subjects who volunteer their time, their effort, their eyes to participate in a clinical trial. And when you think about it, over the last several decades, every major advance in medicine has been made because somebody has sat in a chair and volunteered their time and part of their body to medicine to say, I'm willing to try this, even though I know it may not work. And to me, that was the most moving thing as a physician, to sit with a patient who's suffering from vision loss and who's there. And I have to tell them, look, we've done everything we can do to make this as safe as possible for you. We've We've made it as feasible as possible by testing it. But at the end of the day, I'm not sure if this is going to work, but this is the best we have. And it's amazing to me that we've had 16 people participate in trials here at USC in this particular trial, and hundreds other around the world, and thousands over the last several decades. So the process is truly, truly impressive. And really, the patients we have to thank, because they're 50%, and their families who take a lot of time out of their life to bring these patients to us, to take care of them, and to talk to them through what could be a very difficult decision in their life. So we developed a surgical plan to insert this implant, this 3 by 6 millimeter implant that has 100,000 cells in it, into the area of the eye where the RPE cells, as Dennis described, had died or had disappeared. And we were lucky enough to have 4 very amazing patients. And I remember my first patient that I talked to enroll in the trial, and I said, this is where we are. This is the science and the clinical trial. And she said, I am going to do this. And even if it doesn't work for me, I am happy to help make it work for the next person. And I just walked out of the room that day and I said, this is an amazing world that we live in because of people like her. And so we enrolled 4 subjects. And as you can see on the screen, the results of those 4 subjects, or 5 subjects, were published in Science Translational Medicine. And you can see the results of one of the subjects on the screen there. The implant, which you can see is shaped like a bottle, is underneath the retina in the place that it's supposed to be. And it's actually covering the area of atrophy, because you can't see it anymore in that patient. So we had actually placed it in the correct location. And the results of that preliminary study, and it's still ongoing, so I can't say that it's final at all, but the results were encouraging. One of our patients was able to read 3 lines better on the eye chart that many of you have seen on the eye doctor. And 2 of them were able to show signs on testing that their retina was functioning better than before. And these are patients who've been losing vision for decades, remember. So we're not expecting them to read the 20/20 line on the chart the month after surgery. But those early signs are the beginning of a painstaking process to see how we can translate all this science and all this knowledge into something that's going to help people on a day-to-day basis, to help them see better, to help them read, to help them drive, to help them function in social circumstances where they otherwise don't feel comfortable because they can't see well. And so it has really been my pleasure, my honor to be part of this study and to have patients like we've had and to be part of this amazing team that you see here. So thank you all very much. And Jane Lipkowski from RPT is our next speaker and the sponsor of the clinical trial that I just talked about.
All right, well, thank you very much. And I'm Jane Lipkowski. I am the president of Regenerative Patch Technology, which is the company that is looking to develop this for the eventual disposition and the eventual treatment of patients with macular degeneration. And one of the things that you heard from Amir is that, we've started clinical trials and we're looking at trying to look at both the safety and the efficacy. And it is a very, 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 very standard thing to do right now in looking with the FDA and looking with regulatory bodies to advance those programs, to look not just at the safety and the early efficacy, but to go into more extensive clinical trials, to look at, again, how robust is this treatment, how safe is this treatment, and how can we look at delivering this as a final product, as a final therapy for patients around the world, around the country, and around the world for the treatment of macular degeneration. And again, this is a disease that is very prevalent in our population. There are about 100 to 150,000 new cases of age-related macular degeneration that happen every year in the United States and about an equal number in Europe. Similarly, the prevalence. There are over 1 million patients in the United States and in Europe that have this particular disease. So this is an indication that can vastly affect the therapy can vastly affect and have positive implications for our patients. So what do we need to do going forward? Well, obviously, we need to work with regulatory bodies and work with, our patients to look at scaling up these clinical trials, looking at phase 2 clinical trials, looking at phase 3 clinical trials, so we can register this particular product for eventual widespread use. We need to look at training physicians. Right now, this has been a small study, and there have been really primarily a single surgeon who has been involved in implanting these in the patients in this study. We need to look at expanding that and having multiple surgeons around the country and eventually around the world learn how to deliver properly this implant. Along with what Joe had indicated, we need to look at scaling the manufacturing. We're looking today, we manufacture about 32 to up to 96 implants at a time. But you think about looking at a market that is maybe 150 to 300,000 patients a year, you have to scale up that. And that's something that we need to look forward to. We need to look at getting clearance for our manufacturing. And what Joe indicated was that there is, it's very particular how you manufacture your product. And there is all kinds of stringency that goes along and testing that goes along with the manufacture of that product. That bar goes up as you go towards even looking at marketing this particular product. We have to look at the storage of the product and how we're going to ship it. How are they going to get to all of the clinical trial sites, and how is it going to get eventually to healthcare providers to provide for patients? We also need to look very, very, very importantly at the costs and working towards making this much more efficiently and looking at generating and using pricing that is compatible with all of the various reimbursement policies and the reimbursement so that this can be effective and affordable by patients. And then finally, we need to look at building a sales and technical force, support force, to again, service the patients that we need. So it's really, as I have been working now in this particular field for about 30 years in translating stem cell-based therapies into clinical trials and beyond, it has been my pleasure for the last 9 years to be working with this particular team. So one of the things, again, to finish up with what Amir was saying, is that we really rest upon patients who have been patients and volunteers in these particular clinical trials. And one of the things we'd like to do is show you now a video of one of the subjects who is in our trials, who's been a tremendous pleasure to work with, and someone who has been really the soul and the reason why we do this work. Thank you.
My name is Anna Kuhl. I have lived in Southern California since, my goodness, since I was 22. In the beginning, I didn't notice anything different for a long, long time, except that my vision deteriorated and I needed stronger and stronger glasses. All of a sudden, I saw this big black area, and I was so surprised and scared that I called USC, the Eye Institute, right away and made an appointment for the same day.
Anna Kuhl came to us really looking for an advanced new technique to restore her sight. She had been going blind from a condition called macular degeneration. So what happens in this condition is The retina, which lines the inside of the eye, has light-sensing cells called rods and cones. And in this condition, macular degeneration, the rods and cones get affected in the center of the retina, the part that you read with, recognize faces, and watch television with.
When I realized that I have dry macular degeneration, my vision was still pretty good. But as time went on and I lost these cells.
Then that area becomes atrophic, meaning completely degenerated and destroyed. So you end up with a black blind spot. Wherever you look, there's this blind spot. So if you try to look at somebody's face, there's a blind spot. If you try to read, there's this blind spot.
When I lost the central vision in my left eye, I had to give up many things that I used to do. I became much more dependent on other people.
What we came up with is the concept of just replacing those aged and missing cells using stem cells.
We've gotten to the point where we can differentiate stem cells into actual mature adult RPE, which is the exact cell type that's missing in the patients. So what we do is grow those stem cells into the RPE in the lab on a dish and grow them on this very thin layer of parylene or plastic.
Our innovation has been to engineer a scaffold that is able to carry these cells in and able to be implanted in the eye and seamlessly integrate with the eye, because it really does need to do that latter part.
So it's essentially the cells that are missing in the exact anatomic configuration that they should be in when they're delivered to that space.
After the procedure, my life changed in many ways. I could tell so soon afterwards that something was happening in this eye. When I could see whole faces on the TV, and also the whole face of Hans, my husband, and other people, because I had a very hard time recognizing people before. If you don't recognize people, you don't even like to go to parties or anything, and now I like parties again.
Well, most people would say, what does engineering have to do with the stem cell project? Isn't that just biology and cells? But in this case, it clearly plays a major role, because cells alone could not get this job done.
It's a beautiful combination of engineering, medicine, science, and technology, and it all comes together in this one 3x6 millimeter patch.
My hopes for the future are mainly that these cells, these implanted cells, will stay alive, and that many more people can experience the same as I have and can be helped.
Well, thank you. We can watch it again. The video never gets old. We've seen it so many times. It's what keeps us going, and it gives you a little bit of appreciation of what a difference this technology has made or is starting to make, and what the future could potentially hold. So at this point, I'd like to open it up for questions, and I'll try to pick you in order. So we'll need this mic here, Andy. So we'll start. Andy, why don't you just pass the mic to whoever you think raised their hand first. Please identify yourself and ask your question. Oh, it's not on. Could you give the one, Andy, maybe you have? Okay. There we go.
Okay. Oh, that's better.
Okay.
Sorry. My name is Ann Knight, and I was wondering, I've got two questions. The first is, do you think the stem cells are actually engrafting, or do you think it's more of a paracrine effect that's having the effect? And then secondly, you took the humans and put it in the rat, and I guess you didn't see any inflammatory processes, but could you conversely take another species and use it in human or cross species?
Okay, some two technical questions right off the bat. So for those who are not in the stem cell world, paracrine means that, the question is, did the graft actually, the cells integrate and form a true transplant, or was it because of the presence of the cells and the growth factors they were putting out, did that have a beneficial effect? So that was the first question. And I think I'd like to call on my colleague David Hinton here. I can answer it, but let's have David answer that. We've seen some great results in preclinical studies, if you would mind taking that.
Yeah, that's a great question. And we think it's working both ways. And so, for an example, there's a growth factor that's secreted on the side of the RPE cell facing the photoreceptors that is neuroprotective. And it's secreted at high levels in these RPE cells growing on the paraline. So that would be a paracrine effect. On the other hand, as we saw in the rat retina, those human stem cell-derived RPE integrated very, very well into the rat retina, and there was no associated inflammatory response. So we think it is a real engraftment into the subretinal space where the RPE cells are.
Thanks, David. And so the second part of the question, if I remember, is that, you took these human cells derived, human cells into the rat, and they worked. What did we have to do to make that work? Because it's a cross species. And then the question was, could we take rat and put it into, or could we take rat into pig or rat into human, etc.? So I'll have Dr. Dennis Clegg address that.
Yeah, good question. And to show that this was safe and efficacious, as David described, we did experiments with the human cells, putting them in a rat. And the immune system in the rat would reject those cells as foreign. So we gave the rats drugs to cause suppression of their immune system. Now, in the human case, we're using cells from a different person and putting them into a patient. So we're also immune suppressing the patients, but we only do it for 60 days. We're putting this implant behind the blood-brain barrier, and after 60 days, we think the incision will heal up and they won't be rejected. And so far, that's what we're seeing in the patients.
And to add to that, the eye is, it's a complicated structure, but it's been thought forever to have more immune privilege than the rest of the body. It has to do with how the tight junctions in the blood vessels are and so forth. But having said that, so we might not need as much, or we might be able to locally immunosuppress instead of giving systemic or whole body immunosuppression. But those are things we're going to look into. But two excellent questions to get us started off. Thank you. Yes.
Shamil, founder of Tooth Fairy Stem Cell. I would like to know, have you guys explored injecting the stem cells via hydrogel?
So another technical question. So the question is, you've used this one particular type of substrate, which is what you heard was paraline. We use paraline because it's USP class 6, so it has the highest biocompatibility type of approach. But the question is, could we have used hydrogel? We used hydrogel. The problem is we were having a lot of difficulty getting them to sit on a monolayer and be apically oriented. So it isn't, and there are other people who are using hydrogels. There are also other people who are using PLGA. And we also looked at PLGA and polycaprolactone. But the advantage of the paraline was, A, we could get the monolayer to be situated perfectly on the surface, and B, we could micro machine, and we talked a little bit about that using photolithography, another complicated word, but we were able to micro machine it such that you could get the nutrients transfers, and you could match that nutrient transfer to be exactly like the natural membrane, which is the Brooks membrane, so around 200 kilodaltons. So both because we could
process and machine and verify and validate and get that substrate to be identical and mass produce it, much like computer chips. And because it has so much biocompatibility, we went with that. But other substrates are being explored. So another good question. Andy, all the way in the back, last row.
Is it on? Is it on? Yeah, my name's Ed Ariano, and I was wondering, there's a selective laser how do you pronounce that? With
plasticity.
anyhow, if this procedure were done, would this interfere with something later on with stem cell research?
Okay, so another good question and a technical question, but I think I heard the procedure you're talking about is SLT, which is selective laser trabeculoplasty. And being an ophthalmologist, I can pronounce those words. But having said that, that is a glaucoma procedure applied to the front of the eye, the drainage part of the eye. And so, no, you could have that procedure, and as long as your optic nerve is not damaged and your eye pressures are not elevated, then no, it doesn't exclude you from this therapy, because this is in the back of the eye. The SLT procedure you had was in the front of the eye. You're welcome, Ed.
Thank you. Roger Smith. Are you recruiting now for additional clinical trials?
Good question. Are we recruiting for additional clinical trials? Jane?
So we're preparing for additional clinical trials right now. We're working with the Food and Drug Administration to get approval for those particular clinical trials and looking at putting together the kind of manufacturing that is going to be required in order to expand those clinical trials. So we're actively preparing for those.
So my name is Chris. So I was just wondering, what was the optimal media that you used to develop the RPE stem cells? Like, how did you differentiate them?
How do we differentiate them from the embryonic stem cells? So, Dr. Clegg.
Yeah, so how do we convert that pluripotent cell that can make every cell into just one cell type? And what we started with was to try and mimic what normally goes on in development. Your body, as you grow from when egg meets sperm and you start developing all your tissues, there are growth factors that are released in the vicinity of cells, and it causes them to become a particular cell type. And from studies of mice and frogs and even flies, we know what some of those factors are. So we tried to mimic that in the Petri dish to get those cells to become RPE. And it took a lot of hard work and a lot of different combinations of things. And we now have several methods to make the RPE cells, and we can get them to where they're 99% RPE and don't grow out of control, don't make a bone or a heart in your eye. So it took a lot of work, but we think we have a good procedure now to do that.
Okay. Yes, please.
Hello, my name is Nick Atwater. I have a little bit less technical, maybe more epidemiological question. Is there an increased likelihood for a younger person who's had retinal detachments to later in life develop AMD?
Dr. Kashani?
So, good question. Retinal detachments are a process whereby the retina separates from the RPE. And that can happen spontaneously for a number of reasons. In general, it's not associated with macular degeneration of the type that we're talking about here, which is age-related macular degeneration. But areas of the retina that have been detached and then reattached can undergo other kinds of atrophy depending on what else is going on. So I would say not this particular kind of macular degeneration, but you have to remember macular degeneration is a kind of a broad term and there's many types. And here we're talking specifically about dry age-related macular degeneration.
Mm-hmm. Sure. In the front row here, Andy, I think there's a gentleman here.
My name is James Black. I'm a patient advocate. I've got a question about the shape of the implant patch. And could you talk about how to obtain that form?
Yes, it's a good question. So why does it look paddle shaped or bottle shaped? We had to actually think a lot about that. So there were a number of factors that went through, and I'll share a few with you. One, we needed a handle to hold it, and that handle to get it underneath the retina. So you needed some place to hold it, and so that is where the little, what you would think is the bottle top or the handle of the paddle is. Secondly, the macula is a certain size. This area that we're trying to cover, where there's degeneration, which covers roughly the central 20 degrees of your visual field, and so that is a certain diameter and certain size. So we made it to fit that area. So the paddle to hold it, and the size so we can cover those areas in the macula to give enough coverage so it covers the whole defect. All the way in the back? Oh, no, halfway to the back.
So my name is Pat Corley, and I'm actually a retired nurse from USC. But my question is, the incidence of this particular type of degeneration in the African American population, I was wondering if anyone had any numbers on that. And number two, just to encourage Jane to, when she starts her marketing, to actually market in our community where we actually have access to stem cell and other things that might be beneficial to our community.
You want to take that question about AMD prevalence?
Great question. So the basic answer is actually African Americans are at probably one of the least risks for developing age-related macular degeneration. The primary risk factors, for lots of reasons that we don't understand, are age, actually Caucasian ethnicity, and some dietary factors, and some genetic factors as well. But at least in this regard, it's not one of the leading causes among African Americans.
Yes, sir.
Dr. Homoyo, you guys mentioned that this was primarily focused towards the dry type of age-related macular degeneration. How would it apply, or any other therapies with stem cells apply to the wet type?
Okay, good question. So how would this therapy apply to wet macular degeneration? Recall at the very beginning, I mentioned there were two types, the dry and the wet. And so the wet type means that new blood vessels grow where there shouldn't be. Abnormal blood vessels grow into this area of the macula, and they bleed and leak, and they can cause hemorrhage. Now, we didn't start there, and the reason is the blood, this whole earlier talk about immunogenicity and the eye being immunoprivileged. Well, if there's a blood vessel in there that's abnormal, leaking and bleeding, well, you have a lot of factors that can come in, and it could set up a rejection reaction in our mind. And also, of course, when you're operating and there's a blood vessel, it could bleed and cause a lot of trouble that way. And also for wet macular degeneration, there are lots of injections that we do now that can slow it down. So for all those reasons, we elected to go into the dry. Now, having said that, if it's advanced wet, and it's really caused a lot of bleeding, as I mentioned in London, they're going in, removing the blood and putting a patch of stem cell derived RPE, and we can certainly do that as well. So it isn't that we cannot do it, but it's just that condition is treated initially by injections, which can manage a significant amount of that disease. And secondly, because there's a blood vessel, there's a danger of it bleeding and also setting up an immune reaction. So it isn't that we can't, but we started with the dry macular degeneration for those reasons. So, okay, Kevin's going to give me the hook unless, Kevin, you want one more question? Okay, one more question, and then that's it.
My name is Charles Otieno. I'm a physician. Are we able to use IPS instead of embryonal?
Great question. So another technical question for those of you who are well-versed know the difference between the two cell types. But at this point, maybe I'll have Jane take that question, if you would.
Yeah, in theory, yes, you could also use IPS cells, and there are a variety of groups out there in both Japan and in here in the Los Angeles area, and also in Washington, that are looking at using iPS cells, because you can also look at differentiating them into RPE cells. One of the things that we're very interested in looking at the embryonic stem cells is that we can use one line for potentially manufacturing for all particular people, and that it be much more easy to produce the implant for a variety of different, and the size of the population that we're looking at producing and treating in the future. So thank you.
So before I turn over the mic to Kevin, I'd like to acknowledge one more person who's been really working very hard behind the scenes. Debbie Mitra, please stand up and take a bow. She's been our program manager for the CIRM grant and has done a wonderful job and made sure that we worked our way through this session as well. So thank you again, and here's Kevin.
I'd like to ask for one more round of applause for our extraordinary panel. I think they did a wonderful job turning really complex science into something that even I can understand. So that's an achievement in itself. I'd also like to thank Andy McMahon and his team here at USC, Ching and Christy, for helping make this all possible. I know how much work they put into this, even to the point of getting the sun to go down at a time where we were able to enjoy the slides. So that's a remarkable productivity. But mostly I'd like to thank you, not just for braving LA traffic and giving up a Wednesday evening to come out and listen to a science talk, but for your support for science and for stem cell research in particular. At a time in this country when science is increasingly under attack and when funding for science is increasingly under threat, it's wonderful to see so many people willing to come out and support it. What we've heard here tonight is extraordinary, but this is just one snapshot of so much that's going on here at USC. And even that's just a snapshot of what's going on all around California. There's remarkable work going on around California. CIRM, as I mentioned at the beginning of the show, has funded 55 clinical trials, and everything from heart disease and stroke to rare diseases like sickle cell disease. And we hope with your support to be able to fund many, many more in the years to come. So thank you all very much, and good night.